Hi guys, and welcome to my video, DNA Replication, Part 1 in the Cell. So as you know, in week 3, we cover two different processes of DNA replication, one of which is DNA replication in the cell, which we're going to cover in this video, and one of which is PCR, which I'm going to cover in my Part 2 video. They're very similar in many respects, and in other respects, they're different. And so knowing the difference between each of them is helpful. I like to start with DNA replication in the cell because it's slightly more complex. And if you can understand that one, then PCR actually makes a lot more sense because PCR is just a modification, a simplified modification of DNA replication in the cell. So that's why we're going to start with this. So if you recall from last week, cells have to replicate their DNA right before they go through mitosis or meiosis, because if they don't, then the new cell won't have all of the DNA that a cell is supposed to have. Because every cell is supposed to be diploid. It's supposed to have 2 full copies of every chromosome. And if you split the cell in half, then they wouldn't have enough copies of the chromosomes to give to both cells. So they have to photocopy the chromosomes, duplicate the DNA first, which we do during interphase, so that we can give that to the new, or so that we have a copy to give to both the original cell and the new daughter cell. So this week, we're teaching you how the cell does that. And it's actually kind of a complex process. A lot of pieces come and go, and you get to learn them all, which is great fun. But I think it's honestly a testimony builder to see just how complex the process is and just to know that how often it actually works. Like all these pieces fall into place so often and you don't even know it, but they're what help keep you alive. So it's kind of a cool testimony builder, and I hope you'll keep that in mind as we go through this. Okay, so the chromosome, this big, long, double-stranded piece of DNA here. So my blue lines here represent the DNA. Okay, so you've got your nitrogenous bases going across the middle here, etc. Just imagine they're all there. Okay, so these are the DNA backbones, and you got your nitrogenous bases going across the middle. Okay, well, the first thing we have to do in DNA replication in the cell is we have to separate these 2 strands from each other. DNA is designed so that you only need to know the sequence of one strand in order to know, or in order to figure out the sequence of the other strand, right? And we know that because of the base pairing rules. A pairs with T and C pairs with G, right? So if I know that there is an A on this top strand, we know for sure there has to be a T on the other strand. And so because of the way DNA is designed, I can use one of the 2 strands. I can use one strand of DNA as a template, something that I can read from in order to tell me what the other strand is supposed to have on it. If I see an A on this strand, a T must go opposite of it. If I see a C on this strand, a G must go opposite of it. So because of that, we can duplicate the DNA by separating the 2 strands and then reading each of the 2 strands and building a complementary copy to it, building a copy that pairs up with that strand. So in the end, we're going to go from 2 strands to 4 strands locked into 2 double-stranded molecules. And I'll show you kind of what I mean by that here in a sec. So the goal that the first thing we have to do is separate these 2 strands so that we can read each strand and make a complementary copy of it. That's the goal. To do that, the cell has to break those hydrogen bonds that are across the middle. If you recall, those nitrogenous bases are held together across the middle by hydrogen bonds. Well, the cell has to get those to break so that we can separate the 2 strands going that way. Those hydrogen base pairs don't break at body temperature, which is what we're working with here. So the cell has to get a little creative and go and force them to break. And the way it does that is it uses a protein, specifically an enzyme, called helicase. Now, here's a vocab word for you here. So enzyme. An enzyme is a protein that makes chemical reactions happen faster. So all enzymes are proteins for the purposes of this class at least, but not all proteins are enzymes. Okay, so an enzyme is a protein that makes a chemical reaction happen faster. And the cell has to use a bunch of these enzymes because DNA, if it were just left up to itself, it would never replicate. It would, at best, it would take hundreds of billions of years to replicate itself. We kind of want to duplicate the DNA faster than every 100 bajillion years, right? So we have to use a bunch of these enzymes to help make this chemical reaction happen faster than it would on its own. So we're going to employ a bunch of these enzymes doing various jobs in order to get the job done. And as I mentioned, one of those enzymes is called helicase. Helicase. So helicase is an enzyme responsible for unwinding the DNA helix. The way that I like to think of him is he's like a zipper. If the DNA is like one of those zippers that you've got on your jacket, then the little zipper thing, I don't know, like the little, you know what I'm talking about, right? You got like your little, a zipper, right? And you pull it and it unzips or zips up your shirt, right? Well, that's kind of what helicase is. Usually we draw it as kind of like a little donut-shaped thing or a big blob or whatnot. So this is helicase, but basically it wraps around the DNA and then just starts unwinding the DNA, unzipping the DNA so that we can separate those two strands. He's breaking those hydrogen bonds across the middle, and he's unzipping the two strands, separating them from each other. Well, it turns out that in some of the drawings that you'll see, they show it where you've got just one helicase molecule unzipping the DNA. That's a simplified drawing. What's really going on is you've actually got two of them that hop onto the DNA back to back at this spot that we call the origin of replication. Here's that vocab word here, origin of replication. So these two helicase molecules hop on at the origin of replication. It's just this spot on the DNA that the helicase molecules hop onto. We get two of them back to back, kind of like two zippers on like a backpack or something. You know how you got two zippers back to back and one unzips one way, the other one unzips the other way. That's kind of how this works. So one of these helicase molecules is going to take off going this way, unzipping the DNA, and the other one's going to take off going that way, unzipping the DNA. So after a few seconds, if I erase here a little bit, after a few seconds, we'll have one helicase molecule that's reached over to here, and one helicase molecule that's reached over to here, and of course they're still going. And the DNA in between them will be unzipped. Will look something like this. Now, what we've got going, so notice these two strands, the upper blue strand and the lower blue strand here, are no longer hydrogen bonded to each other. They are single-stranded DNA. So these helicases have separated the two strands, so they're no longer hydrogen bonded to each other, which is good. That's what we wanted it to do. And it will just keep right on going off the page and continue unzipping the chromosome until it either runs into another helicase or falls off. The end, right? So over time, we will unzip the entirety of the chromosome. But while that's happening, we've got a couple of other proteins and enzymes that are going to come in here and help us out. One, we've got a problem here. DNA, these DNA strands right now are single-stranded. DNA hates being single-stranded. It absolutely hates it. The reason for that is in week 2, you learned about those nitrogenous bases that poke out across the middle. They hate being with water. They hate touching water. The phosphates and the sugars, the backbone, they're okay touching water, but the nitrogenous bases, they're hydrophobic. They hate touching water. So the reason why DNA is double-stranded in the way that it is because it's in that helix and stuff. The sugars and the phosphates can touch the water, and the nitrogenous bases can be protected on the inside and kept happy away from the water. That's fine and dandy until we separate these two strands. Now those nitrogenous bases are just poking out here into the water, and they don't have their fellow strands to help protect them from the water. And so they're unhappy. They desperately want to be double-stranded again so that they can be protected from the water again. And so naturally, this bottom strand here and that top strand there will want to magnetize back together again right behind the zipper. So helicase would unzip the DNA and then it would rezip itself right behind the helicase. That sounds like the most obnoxious zipper of all time, right? You unzip your jacket and it rezips right as you're going. That's no fun. So we want to, in order for this to work, we need to separate the two strands and keep them separated long enough for me to actually copy them. So to prevent them from going back together again, we have a little protein here. We're going to get a bunch of copies of him. We just have a bunch of this particular protein called single-stranded binding protein. Ain't that creative? Single-stranded binding protein. Or sometimes we abbreviate him SSB for short, for single-stranded binding. But his name describes exactly what he does. He finds single-stranded DNA and he sticks to it. That's his job in life. That's all he does. So he's not an enzyme. He's just a protein that finds single-stranded DNA and sticks to it. The reason why that's important is the single-stranded binding protein sticks to the DNA and prevents it from sticking to the other strand. So his job is to basically keep these two strands separate from each other after helicase has unwound them, so that they don't just stick right back together again behind the helicase. So we will have bunches of these single-stranded binding proteins just coating both strands of the DNA, and more will keep adding on as the helicases continue to unzip more. And their whole goal is they're just going to sit there and prevent the two strands from going back together again. Okay. One other protein that I want to add right here. Turns out that we've got another problem in front of helicase. So there was a problem behind helicase with the two strands trying to come back together again. Now we've got a problem in front of helicase. And to best show you this, you kind of need to see a picture. So I'm going to pop over to my website here. See here. Here we go. Okay, here we go. It turns out that if you've ever taken two ropes, you wrap them around each other, coil them up or whatnot. In this case, it looks like a rubber band, and then you try to pull them straight apart. So not unwind them, just pull them straight apart like this. You'll find that you can only pull them apart so far. And ahead of it, it becomes super, super tight. And you start getting these little kinks in the rope like this. You see that? That is called supercoiling. That's the scientific term for that, where it becomes more twisted, more tightly coiled ahead of it, and you get those little kinks. And just like how at some point you can no longer pull the ropes apart, this actually happens the same in DNA. When helicase goes in to try and unwind those DNA strands, it turns out that in front of it, the DNA actually supercoils just like with these ropes, and it starts kinking. And the helicase gets stuck. Just like you can't pull those strands further apart, the helicase at some point gets stuck where he can't get past those kinks. He can't get past the supercoils. And so somehow we've got to fix that, because we can't be having helicase getting stuck all the time. So there is an enzyme who's going to hop in here and fix that problem. And he's got, in my opinion, one of the best names in this whole week. His name is topoisomerase. Ain't that a tongue twister? Topoisomerase. Name your kid that. So topoisomerase is responsible for fixing that supercoiling. So he actually hops on the DNA in front of each helicase. So he's traveling along in front of each helicase as it's unwinding the DNA. And he's actually fixing the supercoilings to make sure that helicase never gets stuck. And you don't need to know the process for how he does that, but his whole job is he's traveling in front of helicase, making sure to undo all of the supercoiling so that helicase can unzip the DNA successfully and never get stuck. So that's topoisomerase's job. So he travels in front of helicase there. All right. So far, we've got 3 proteins on the map. We've got helicase unzipping the DNA, topoisomerase undoing the supercoiling so that helicase doesn't get stuck, and single-stranded binding protein preventing the 2 strands from going back together after they've been separated. Okay. Let me erase this here to make room. So before I move on, let me point out a couple of features here in the DNA. If you'll notice, behind each of these helicases sort of is making like a fork in the road, you might say. Like a fork in the road kind of looks like this, right? So you've got a fork in the road. And notice this helicase is doing the same thing. So it's taking the DNA and it's splitting it into like this little fork in the road sort of thing. So these are what we call replication forks. So the replication fork is kind of the split, the Y-shaped area, the split right behind helicase, where helicase is unwinding the DNA. It forms this replication fork. If you notice, since we have 2 helicases for every origin, then we're going to have 2 replication forks, one behind each helicase. And if you connect these 2 together, we form this structure here in the middle that we've had, we call that a replication bubble. So 2 replication forks equals 1 replication bubble. But yes, for every origin of replication, there are 2 replication forks, and then those 2 replication forks make a replication bubble. All right. So that's what those terms mean when you see them. Back it up here so that we can resume our story. So we have finally unzipped the DNA strands. Yay, right? We have finally accomplished our first goal of separating those 2 DNA strands. So now we need to construct a new strand to pair up with each of these 2 strands here so that we can get 2 complete DNA double helixes when we're done. So the enzyme that does that, because DNA doesn't just self-assemble, you have to have a construction worker, an enzyme, come in there and assemble the new strand. His name is DNA polymerase 3. And we usually write it with a Roman numeral 3. That's quite a long name there, huh? And at this point, I'll pause and teach you a way, because you're probably getting a little overwhelmed with all of these different enzymes, because you will have to remember them and what they do. So let me give you a little trick here that helps with at least a few of the enzymes in remembering their names and remembering what each enzyme does. Turns out that when we scientists name enzymes, there's actually some rules behind it. There's logic behind how we name enzymes, believe it or not. And so if you understand those rules, then it can kind of help you remember the names. So anything that ends in the suffix ase, ACE, is an enzyme. So ASE means enzyme. And then anything before the ASE either says what the enzyme does or what it's associated with. So DNA polymerase 3 is an enzyme that builds a polymer. A polymer is just a chain. So it's an enzyme that builds a chain of DNA, which is exactly what the enzyme does. It's going to read the template strand, read the dark blue strand, and build a complementary strand to go with it. So it is building a chain of DNA. That's exactly what it says, what the name says it's doing. The 3 here at the end just means that it happened to be the third enzyme that builds a chain of DNA that we discovered. So it is DNA polymerase 3. So he's going to be the one responsible for building the new strand. However, DNA polymerase 3 has a weakness. His weakness is he doesn't know how to start a brand new strand. He only knows how to make a pre-existing strand longer. That kind of seems like a catch, huh? Because isn't he supposed to be the one building the new strand? So how do we solve this chicken and the egg scenario? He's got to build the new strand, but we have to have at least a bit of a new strand already down for him to be able to go finish it. So we have to have yet another protein pop in before DNA polymerase 3 in order to satisfy this particular weakness. This protein, it's an enzyme called primase. And primase is going to come in here onto the DNA strands. And it's going to, I'll move this single-stranded binding protein here. It's going to build part of the new strand. So right there, so that would pair up right there. Are you following me there? So it's going to build part of the new strand. But the nice thing is now that means we've got part of a new strand. So there is now a pre-existing strand that DNA polymerase 3 can make longer. So primase doesn't lay down the entire new strand. Primase lays down between 10 to 30 base pairs, something in that vicinity. I'd have to go double check what the exact numbers are. It varies. There's some number for in the cell and some number for outside the cell. So you'd have to go double check to see what the exact primer lengths are. I think it's 10 to 30. Oh, wait, no, 10 to 20, something like that. I'd go look if I were you, because I don't have those memorized off the top of my head. But anyway, it lays down just a little piece of the second strand here. The other thing to note is this little piece is not made out of DNA. Primase builds it out of RNA. Ain't that weird? RNA, we're going to learn about RNA more in week 5, but RNA is basically a cheap, chintzy version of DNA. So it's easier for the cell to make RNA, but RNA also isn't as durable. It doesn't last very long. Whereas where DNA can last for thousands of years, RNA can last maybe a couple of days if you're lucky. So we don't normally build, we're trying to build this out of DNA. That's why we're building out of DNA, but primase only knows how to work with RNA, and it's his job to build the primer. And so we settle for the fact that the primer is made out of RNA, and later on we'll have to fix that fact. We'll have to get the RNA out of there. But for now, we have to build the primer out of RNA. So he's going to lay down an RNA primer right there, and we'll also lay one down here on the bottom. Okay. And now that there is a strand here, these little pieces, we call them primers. Primer, hence primase, an enzyme that builds a primer. And the reason why it's called a primer is when you prime something, that means to get it started, to get it going, like primer paint. You paint the wall with primer first, and then it helps the real paint stick better. Oops, sorry. Sorry about that. It helps the real paint stick better. Same thing with priming a pump. You may have heard that phrase. You add a little bit of water so that we can get it started and get it going. So when you prime something, it gets it started, which is what primase has just done with the RNA primer here, is it started that new strand. So now DNA polymerase 3 has a strand that it can now make longer. So it hops on at the primer. Here will be my little DNA polymerase. So we're going to hop on at the primer. And then it's going to start traveling and reading the DNA template, the dark blue strand, and putting in the complementary base opposite of it. So if you've got an A on one strand, then you're going to put, he's going to read an A there and put in a T on the new strand. And then if he sees a C over there, he'll put a G in on this strand. Now, an important point to make here, and I'm going to switch over to another screen to help make it, is this is DNA polymerase 3's second little, it's very nitpicky about this. So the first one was it can't start a new strand. It can only make one longer. The second one is it can only build in one direction. You remember how in week 2, we learned about how DNA has directionality. There's the 5' end and a 3' end, right? So like if this was the 5' end and the 3' end, and then of course it's anti-parallel. So this is the 5' end over here, the 3' end of the dark blue strands, right? Well, DNA polymerase 3 can only build in the 5' to 3' direction. Now, the other way that you may hear that, and this confuses a lot of students. I think it's in the Khan Academy video that's in your reading. The other way they say it is that DNA polymerase 3 can only add to the 3' end. Now, it may seem like those two statements, building 5' to 3' and only adding to the 3' end, some students get confused because they're like, wait, but those aren't the same thing. Actually, it is. And let me pop over to another screen here to show you what we mean by these statements. Okay, so here's just a really short piece of DNA that I just wrote out here. It's just a random piece of double-stranded DNA, right? So this top sequence here is the top strand. This bottom sequence here is the bottom strand. I've labeled the 5' and 3' ends of both strands, okay? So let's pretend helicase comes in here and it separates these two strands like this. All right, so now let's say I'm trying to copy this top one. So let's say we laid down a primer somewhere here off screen, okay? We're gonna pretend we've laid down a primer, ignore it, okay? So polymerase is gonna hop on, and it always builds in the 5' to 3' direction. Build from 5 to 3. And that is from the perspective of the strand being constructed, okay? That is a common point of confusion for students. It is always from the perspective of the strand being constructed. So, for example, if I'm going to try and build the complementary strands to this top blue one, a lot of people would say, well, if you're building 5 to 3, then you will build left to right, because the 5's on the left, the 3's on the right. You won't though, because it's from the perspective of the strand being constructed. And this is the 5' end of the strand being constructed. And so you would actually, for this top strand, or for copying this chunk up here, we're going to build right to left, because we're going to build in the 5' to 3' direction. So I would lay down this base, and then this base, and then this base, and then this base, and then this base. So notice as he goes along, he's building in the 5' to 3' direction, away from the 5 toward the 3, because this would be the 3' end of the new strand. And then notice that the DNA polymerase 3, all he has to do to know what to build into this strand is read the base on the template strand and follow the base pairing rules. And there are just individual bases just floating around in the liquid. So the polymerase will stop here and be like, okay, there's a G right there. Okay, I need a C, got one. And then he adds in a C. Then he shifts over one. He's like, okay, there's a T there. So I need an A, got one. So that's what he's doing. And he's building in the 5' to 3' direction from the perspective of the strand being constructed, which means your read 3 to 5. Because notice in this case, we are traveling right to left. From the perspective of the strand being built, that's from 5 to 3. From the perspective of the strand being read, that's from 3 to 5. So don't get those mistaken. You read 3' to 5', you build 5' to 3'. So the 5' to 3' rule is from the perspective of the strand being constructed. I've repeated it like 7 times, so hopefully you won't forget. Okay. So then notice down here, if we're going to try and copy this bottom strand, notice the 5' end of the new strand is right here. So down here, we will be building from left to right. So this would be an A, and then you would go T, and then T, and then C, and then G, and then G, etc. Okay. But so the moral of the story is polymerase can only build in the 5' to 3' direction. And when we say that, that's what we mean, is that the strand being constructed can only build. Now, I told you I would clarify that Khan Academy statement of that you can only add bases to the 3' end. That's exactly the same thing. See, if this is the 3' end of the strand, then if I added a base to the 3' end, I would add this one. And if I added another base to the 3' end, it would be this one, right? Notice I added those 2 bases onto the 3' end of the growing strand, which meant we got 2 bases longer going that way, which is the same thing as 5' to 3'. So saying that you add bases onto the 3' end is the exact same thing as saying you build from 5' to 3', because both statements say you're traveling away from 5 and toward 3. Okay, so there you go. So whichever way you think about it, that's what that means. All right, so back to our story. So if DNA polymerase 3 hops on at that little RNA primer there, we need to ask ourselves, will he travel left or will he travel right? Well, this over here on the left side, that says the left side is the 5' end of that template blue strand up there, which means this is going to be the 5' end of the primer, right? Of the new strand. So polymerase will hop on and it will build the new DNA strand from the end of this primer, and it will build this way. And in the process, he will actually knock off those single-stranded binding proteins so that they're no longer there, because they're no longer needed. The DNA is now back to being double-stranded during that chunk, and so it's no longer tempted to go and stick to its neighbor. So as soon as the polymerase is going in, adding these DNA bases, it's knocking off these single-stranded binding proteins that are no longer needed anymore. So he's just going to hop on at that primer and just come and build the DNA strand. He'll just travel along like a choo-choo train, and he'll just build the DNA strand in the 5' to 3' direction. Okay, and then if he hopped on down here, so if we had polymerase hop on at that primer, notice that the 5' end of the template strand down here is over here on the right. So this would be the 5' end of this primer down here. And so down here, we would build going this way, away from the 5 towards the 3, and knocking off our single-stranded binding proteins. So there we go. We're accomplishing our goal. We're building these DNA strands, and this polymerase will just keep following that helicase, because notice he's traveling the same way helicase is. This guy up top is traveling the same way that helicase on the left is. So they'll just keep right on building those strands and following helicase to the end of time until helicase falls off or bumps into another helicase or whatnot, and then polymerase will finish up over there. And that's fine and dandy and great. But now we have another problem. What about this DNA over here? We didn't copy that DNA or this DNA. And we can't just like turn around on the primer and go that way, because if we were to hop on at this primer and go this way, we would be building in the 3' to 5' direction from the perspective of the strand being constructed, which polymerase 3 can't do that. It can only build in the 5' to 3' direction. So how do we solve this predicament? Because we have to copy the entire chromosome. We have to make a photocopy of the entire length of the sequence. And right now, everything that's before the 5' end of the RNA primer isn't getting copied. It's kind of an issue, huh? So the solution to that problem is primase is going to come. So the primase, that enzyme that we bumped into earlier that built the RNA primer, he's going to come back. And he's going to build another RNA primer right here and another RNA primer right here, both of them right behind the helicases. And then DNA polymerase 3 will come back and it will hop on at the new primer and build from that primer in the 5' to 3' direction until he runs into the back of the next primer, and then he'll just connect it to the back of that primer. Same thing over here. And again, we will knock off those single-stranded binding proteins because they are no longer needed. Okay, knocked off the single-stranded binding proteins. Let me see if I can patch some of my little holes here in my drawing. Okay, so yay, right? We've duplicated those strands. But now we've got yet another problem, because this polymerase up top here, it's traveling left, right? And so it can follow the helicase, fine. But I'm going to lay down this primer right behind the helicase and build from there to there. But while I'm doing that, this helicase that's on the left here is going to keep traveling left. It will go off the screen and keep unzipping more DNA. So that will mean there will be more uncopied DNA, and it will once again be before the 5' end of the primer. So primase, and we're going to have the same story over here, but up on the top strand here, helicase will keep unwinding. So we're going to have more uncopied DNA up top that DNA polymerase 3 can't reach. So primase will have to come and lay down yet another primer, and then we'll build from that to there. And we're just going to keep repeating this little fragmentary construction thing over and over and over again in order to fill in the gaps. So this is where we're going to pause and make a point with some vocab terms. If we were to draw a line down the middle here of at the origin of replication where the two helicases started, now let's just focus on this helicase on the left, okay? So pretend this helicase on the right doesn't exist. Let's just focus on the helicase here on the left. Notice that from the perspective of the top strand in this case, helicase is traveling 5' to 3', right? Because it's traveling left. And for the construction of the new strand, from the perspective of the new strand, this is the 5' end over here. This is the 3' end over here. So it's going this way, helicase is going that way. So the polymerase only has to hop on the DNA once, and then it can just build the new strand in one big, long, continuous chunk as it follows helicase to the end of time. That strand, the strand that you can build in one big, long piece, is called the leading strand. Sorry, that says strand. Leading strand. The leading strand is the one that can be built, or the word they sometimes use is synthesize. That's a fancy word for build. It's the strand that is built in one big, long, continuous piece. And the reason why it can be built in one big, long, continuous piece is because polymerase is traveling in the same direction as helicase. So polymerase can just follow helicase to the end of time. This bottom strand, in this case, however, can't be built in one piece because it has to be built in chunks. The reason for that is down here, building 5' to 3', polymerase is going that way. It's going right? Because there's the 5' end of that primer and the 3' end over here, right? So it has to travel towards the right, but helicase is still going left. So in the case of this bottom strand, helicase and polymerase are traveling in opposite directions. So, and since polymerase can't build on anything that helicase hasn't unwound, that's why we have to build it in fragments. So that's why we have to lay down a primer here and then build, and then helicase will have exposed more, so we'll lay down yet another primer off the screen and then build, and then we'll lay down yet another one and build, and we have to build it in chunks. This strand that we have to build in segments is called the lagging strand. because it's kind of lagging behind. It takes a little bit more effort to copy the lagging strand than it does to copy the leading strand, because DNA polymerase 3 can only build in the 5' to 3' direction. So that's what the lagging strand is. And we have a term here for this little segment, because you'll notice the top strand here, our leading strand, is built in one big long continuous piece. It's got one primer, it's built in one long continuous piece. Down here though, we've got kind of this chunk of DNA located between the two RNA primers, right? You see that? Because we built from the front of this primer to the back end of that primer. And then again, we would do it again before that. So we would go from some primer off the screen over to the back of that primer, et cetera, right? After helicase kept unzipping it, things like that. So the lagging strand is made in these little chunks where you got RNA primer, big long DNA chunk, RNA primer, big long DNA chunk. These big long DNA chunks between the RNA primers, let me erase this to make more room here, are called Okazaki fragments. Named after a Japanese guy and his wife that discovered them. And notice these Okazaki fragments are only found on lagging strands, not leading strands, because the definition of an Okazaki fragment is that chunk of DNA between two RNA primers. And you don't have two RNA primers on the leading strand, you just have the one RNA primer, and then it's built in one big long DNA piece. So Okazaki fragments are only found on lagging strands, because we have to build lagging strands in chunks. The other thing that you may notice here, I have some students that are like, wait a second, and they realize that since there are two replication forks going on here, notice that over here on the right, it's the bottom strand where polymerase is building in one big long continuous piece, and it's the top strand that we're having to build backwards in chunks. So over here, this is the lagging strand. And this is the leading strand. So notice, the top strand isn't always the leading strand, and the bottom strand is not always the lagging strand. It always depends on which helicase you're following. From the perspective of which helicase are you looking at? So on the right side of this origin, in my particular example, the leading strand is the bottom one because the helicase on the right is traveling the same direction polymerase is. Whereas on the left, it's the top strand that's the leading strand because that helicase on the left is traveling the same direction the top polymerase is. So it's not, so the thing to memorize isn't the leading strand is always on top and the lagging strand is always on bottom, because in reality, both the top and the bottom strands are both leading and lagging at various times or at various points along the length. The moral of the story is, from the perspective of one of the helicases, which strand am I constructing in one big long continuous piece? That's the leading strand. Which strand am I building in multiple chunks? That's the lagging strand. Okay. All right. Let's see if we can clean this up again and continue our story. It's up here. Okay. That's backed up probably far enough. Yeah, let's see. Okay, here we go. So we have constructed the DNA strands, right? And we've gone in, we've laid down primers, the helicases have unzipped off the screen, right? We've laid down the primers, etc. So we have completely copied, polymerase 3 has finished his job. He's finished copying both strands, okay? Let me remove our little helicase, or our little polymerase 3 friend there, still sitting there. Put in my primer again. Okay, there we go. Okay, so now we've got yet another problem. There are just so many problems here, aren't there? So our next problem to solve is, remember how I told you that RNA is very fragile? Well, we don't want these little primers that are made out of RNA, we don't want them to stay there because RNA is fragile and it's chintzy and we will get mutations super easily. It will break, it will do all sorts of bad things if I leave it there for too long. So we don't want that RNA to permanently be part of my DNA strand. I want to have double-stranded DNA, not this little RNA DNA hybrid thing that we've got going on over here. So somehow we have to swap out those RNA primers for DNA bases so that it's more longer lasting. So we're going to bring in the next enzyme, and his name is DNA polymerase 1, not to be confused with DNA polymerase 3 that we were using earlier to read the template strand and build the new strand. So don't get those two confused. We've got two DNA polymerases going on here. We've got DNA polymerase 3 that was reading the template strand and building a complementary strand. And now you've got DNA polymerase 1 again with a Roman numeral. His job, he actually has several jobs, so we're going to see him repeatedly here at the end. His first job is he comes in and he actually cuts out the RNA primers. He like physically goes in and chops out all the RNA bases. Okay, so now we've got these gaps in the strand. And remember that this has been built from here off, right? And this was built off this green, and this was built off this green, and we built from here up to there, okay? So pretend the helicase and topoisomerase are gone, all right? Okay, so now we've got, so we cut out those RNA primers. Yay, we no longer have RNA. Good job, DNA polymerase 1, right? You got rid of the RNA. But now we've got another problem that just caused, is now I've got holes in my DNA line. There's gaps, there's these big, huge gaps that were left where the RNA primers used to be, and we cut them out. So holes are just about as bad as having RNA in there. So we got to fix the holes. So DNA polymerase 1 cleans up his own mess. So he was the one that cut out the RNA primers. He is also the one that is going to come in and fill in the gaps with DNA, building in the 5' to 3' direction. So he's going to fill it in there, and he's going to fill it in right there, and he's going to fill it in right there, and he's going to fill it in right there with DNA. So woohoo, we filled in the gaps with DNA. Now it turns out we have yet another problem that's kind of invisible at this point, unless you look really closely and use your imagination. Turns out that for reasons you don't need to know for this class, and if you want to know, you're welcome to talk to me. You can email me or text me, because you don't need to know for this class, but I think it's a fascinating story. For some reason, DNA polymerase 1 cannot make the last little link here. So notice here, I deliberately left like a little gap there at the end. I kind of didn't do it up there, did I, huh? Whoops. But they are, for a reason that you don't need to know, DNA polymerase 1 cannot link the last base in his little DNA fill-in primer to the next base in the actual DNA strand. He can lay down the bases, but that last base, he just can't build that sugar phosphate backbone. He can't take the phosphate of the last base in the replacement primer and hook it to the sugar of the next base. He doesn't know how to do it. Again, there's a reason for it. You don't need to know. So we've got this nick in the backbone is what they sometimes say it as. We've got a phosphate not connected to the next sugar. There's a break in the backbone here, and that's a problem. Broken backbones are not good. So somebody's got to fix that, and DNA polymerase 1 doesn't know how to. So we have to introduce our last enzyme. Aren't you excited? Yeah, this is the last one. His name is ligase. Ligase comes in here and he doesn't add any new bases. Remember, all of the bases have been laid down by DNA polymerase 1. All he does is he takes that phosphate and that sugar and goes click and fixes the nick so that we've got one big continuous backbone. Yay, the backbone is fixed. So he just comes onto the ends of those filled in primers and just goes click and connects them together. So he links the Okazaki fragments together is a way that you sometimes may hear that, because you cut out the primers between the Okazaki fragments and then we fill them in with DNA, and then his job is just go click and you hook those pieces together. So now we've got a full DNA strand without any breaks in the backbones, which is exactly what we're looking for. And had we continued going, you would notice that we now have a piece of double-stranded DNA up top and a piece of double-stranded DNA at the bottom. And because of the DNA replication, or because of the DNA base pairing rules, notice that both of these DNA helixes will be identical to each other, because all you need to know is the sequence of one strand in order to figure out the other one. So the polymerase read this dark blue strand and filled it in with the complementary bases using the base pairing rules. And then it can come and read this bottom strand and do the same thing. And notice, if you compare these two DNA double helixes, they're identical to each other, because each one told us how to read the other, or what the sequence of its missing twin was. And so you wound up with two identical DNA double helixes. We're done with DNA replication in the cell. So now, if you go back to week 2, you have now gone from an I chromosome, or I-shaped unduplicated chromosome, to an X-shaped duplicated chromosome. So each of these two new duplicated helixes would be connected to each other at the centromere. Now you can go through meiosis and you're done there. So that's the big process with DNA replication in the cell. So yeah, there's a lot of enzymes to keep track of. I told you that DNA polymerase 1 has 3 jobs and we only talked about 2. He comes in at the end after all of DNA replication is done and edits the DNA. So he scans along the DNA checking for typos. And there are ways that he can detect a typo. For example, we learned last week that if you pair a purine with a purine, you'll get a bulge in the DNA because it'll be 4 rings across instead of 3 rings, right? So if polymerase 1 is traveling along and he hits a bulge, he's like, oh, that's a problem. And so he can fix it. Same thing with a dip. If you put 2 pyrimidines together, you get a dip in the DNA strand. So that, because DNA polymerase 3, when he's building these strands, he's laying down the new bases at 1,000 base pairs, or at 1,000 bases per second. That's super fast. If we were typing only 4-letter words, that's 200 words per minute, 250 words per minute. I can't type that fast. But so he lays them down really fast, and he makes a typo about 1 every million base pairs or so, which is super crazy that he only makes that many typos. But we'll learn in week 5 that typos in DNA are bad. We don't want typos. We want to make sure that the DNA stays the same as much as possible. So if polymerase, when he's copying here, accidentally puts in a G here where there's supposed to be an A, then DNA polymerase 1 will come back in here and be like, wait a minute, a T is not supposed to be paired with a G, and it'll actually swap that, or cut out that G and replace it with an A, so that it fixes that typo in the strand. So that's its last job, is it edits the DNA. After DNA replication is all done, DNA polymerase 1 comes back through and just double checks everything to make sure we didn't get any typos. And DNA polymerase 1, just like every editor, is imperfect. And so he still occasionally misses one. That's how we get genetic diversity, as we have mutations accumulate in the population over time, but he's really good. He catches most of them. It's very rare that you actually have a mutation that gets missed. So that's really cool. And again, we'll learn more about mutations in week 5. But so that's DNA polymerase 1's third job. So let me know if you have any questions about how this DNA replication in the cell process works. In my next video, I'll talk about how PCR works, and we'll compare it to this process here and show you what's the same, what's different. And please let me know if you have any questions. Email me, text me, set up a tutoring appointment with me. I'm happy to help in any way that I can. All right. Have a great day. Thanks for watching.